Hello, 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 Bio 115 students. It is a pleasure to be with you again. As you know, I'm your instructor, Karen Klein. That was Pearl snoring. Ha. Nothing like a pug. Anyway, this is another lab for you. And this one's going to be a little different if you're using these videos. This one will be a little different than the other ones, but that's okay. We'll just go right along with the procedures. As you are aware, as you've done so many times before, your printout is uploaded in this week's folder, so I'm sure that, as you know, you already have it uploaded and you're looking at it, you're going back and forth between the video and this, or maybe you've printed it out, and that is what you're going to be uploading to the submission link when all is said and done. Now, one of the basic pieces of equipment that biologists use is called a microscope. A microscope is a series of magnifying lenses that are meant for you to be able to, the user to be able to see things that they normally would not be able to see with the naked eye. This is a little bit challenging in a virtual setting. In a real-life situation, you will absolutely be asked to handle a real microscope, and but the simulation does its best to try to repeat that experience. So go ahead and take a look at your handout. That's on page 2. That's an idealized schematic of what's called a compound light microscope. There are many different types. And you see where the eyepieces are. That's where you look through the microscope. Now, generally, the magnification of those eyepieces has a magnification of 10x. So if you were to remove those eyepieces, which I don't recommend doing on a real microscope, don't take the microscope apart, you would be looking at something that's 10 times bigger by looking through them. But they are combined with what we call objective lenses that are further down on the microscope that are on that revolving nose piece. And those objective lenses, usually there are 3 or 4 on a good microscope, and they in and of themselves have magnifications. When combined with the eyepiece magnification of 10 gives you a larger magnification. Usually the lowest objective lens is 4x. So when you're looking through the eyepieces, that would give you a combined magnification of 10 times 4, which is 40 times bigger than the object itself. The next higher magnification on the objective lens is 10x, which gives you a total magnification of 100. Then you have a 40x objective lens, which would give you a total of 400 magnification. And then to look at bacteria, generally there's a 100x, which gives you 1,000 magnifying, total magnification of 1,000 times bigger than your unaided eye. So that's pretty cool. Now, in a real life situation, we would, I should point it out on this, the objective lenses, you can't, the ocular lens you can't see. These are your objective lenses. We have 100, we have 4, there should be 104. There should be one. It's not letting us look back here what it is. 100, 4, and 10. Okay, very good. Here's your stage. That's where the microscope slide, the slide itself. It's a piece of glass where the object is placed upon. Objects that you use a compound light microscope for have to be translucent. You have to be able to see through them because light comes up through the bottom of the scope and through the object placed on the glass slide and then into your eye. We have focus lenses where you move the stage up and down in order to focus on what you're looking at. And then you have stage clips which move the object left or right or front or back. So you don't ever have to touch the slide after you put it on the scope. So if you scroll down to the exercise, you see pages 3 through 3 and 4 all go through the different procedures as far as how to use the virtual microscope. Now, if you are watching this video, it means you have a slow internet connection and you're not doing the virtual lab itself. So you don't need to look at that for the filmed version of this particular lab. Now, I'm going to go ahead and I'm going to put the slides on the scope, and we'll look at the view screen to see what they have available to us. But the parts you are to label, and the images that I have of what of what we're supposed to look at through the virtual lab are different. I got these off of Google Image because I wasn't able to really put a screenshot of the virtual lab into the lab itself. So just so you are aware. So you're to label these, and that's what you wind up uploading to your submission link. So we'll go ahead and we'll get started with the virtual lab of this. But this is what you want to label. You want to label figure 1 on your handout. The first one is on page 5, and it is an amoeba, which is a single-celled protist, they're just really cool. They live in water, and they have those really cool pseudopods where they just kind of shimmy their way around the water, and when they want to consume something, they go ahead and they kind of wrap their pseudopods, their slimy stuff, their cytoplasm, around it and form a vesicle, and then they ingest it. It's pretty cool. So here's our amoeba. Hey, there it is. That's pretty neat. And what we're going to do is we're going to focus. We're at 10x. They want us to look at 100 total magnification. That means that our objective lens is in place of 10, because our ocular lens, as you recall, is also 10. That gives us 100x. And we're going to focus. Now I'm doing the things you would do if you were actually signed in and doing the... There we go. It's a little frustrating. I have a pretty good internet connection, but it's still, I think it really wants you to mess around. Notice how I'm taking responsibility off of me. Yeah, it just kind of went out of focus again. Okay, there it is. Hey, here's your amoeba, and you see this big dark stained circular entity is the nucleus, and you see here's your cell membrane, and here is your cytoplasm in here, and you kind of see pseudopods. Pseudopod, pseudo means fake, pod means foot. So they're not feet, but they kind of look like feet, because that's how that little bugger gets around. It just extends its pseudopods. Pretty cool. All right, so you want to go ahead and label that in figure 1. Or no, not figure 1, right? It's a figure, no, it is figure 1. Okay, on page 5. All right, next we're going to look at Spirogyra. This is a type of algae, and we'll look at it under the microscope, but as you see there in figure 2. All right, we're going to put our amoeba back. And good Lord, I have to sneeze. I am so sorry. This is live, folks. If I sneeze, I sneeze. All right, here's your Spirogyra. Hey, there it is. There you go. I don't have to sneeze anymore. Okay. Nope. And okay, it's at 100x. And it's like a filamentous algae. Like if you're in a pond and you see what just looks like long strands of green hair and a clump, and a big old slimy clump, that's Spirogyra. So here, you have cell walls. Your nuclei are really pretty tough to see. You can see the cell membranes right here. Your chloroplasts are really kind of dark little circles. So it wants you're to measure the cell, or you're to label A, B, and C on figure 2, a cell wall, nucleus, and a chloroplast. So we'll go ahead and we'll have you do that. Next is human cheek cells. Now I alluded to it, but I didn't mention that in the wet lab, like in the real live lab, you actually do look at all this stuff under a real microscope. You look at your own cheek cells. It's pretty cool. It's cool when we do the wet lab, sometimes students find it to be anticlimactic because they're expecting, I don't know what they're expecting. They just kind of look like sunny side up eggs. sunny side up eggs, and kind of the analogy to that is I had a lot of caffeine today and my hands are shaky. I'm trying really hard to. There we go. Sunny side up eggs, meaning in the middle, like in each of these cheek cells, unless you're an android, there should be a nucleus. And then you wind up having, like what I would say would be the yolk of the egg, kind of small. Then you have what I would say like with the egg white, right? That's your cytoplasm. And on the outside there should be, or there is, a cell membrane, but it's pretty thin, so it's hard to see. So you're going to want to label that A, B, and C. And then last but not least, we're going to look at bacteria. Bacteria are teeny, teeny, tiny. And you won't really be able to see, like, oh, whoops, we don't want to take the microscope away. Sorry, guys. They're so tiny that you're not going to really see a bunch of different structures on the inside, not with a compound light microscope. You would need an electron scanning microscope, which we don't have in our school, let alone in our lab, let alone in our virtual lab. I changed the objective lens to 100, which gives us a total magnification of 1,000. And even at 1,000, that's all you're going to see. This looks like some dust. Come on. There we go. And so I'm not going to have you in figure 4, I'm not going to have you label I'm not going to have you label the parts because you can't see the parts. You know, there's a nucleoid region, there's a strand of DNA, there's a cell wall. You can't see them anyway. So what the question asks you, of the 3 different categories of shapes, remember those? Spirochetes, that's the one. What are the other ones? Cocci, and what's the other one? You have to determine in figure 4, what shape are those bacterial cells? All right, so fill that out. That's number 4 here on part 4, bacteria. And you want to fill that out and then submit that to the submission link for this week's lab points. All right, everybody. I hope everybody's well and take care, and we'll see you for the next lab. Bye, guys.